Well, welcome back to the second session this morning of our discussion of the possible role that solvents other than water may have played in the origin of life. We're going to switch a little from planetary science to some real chemistry. We have Nicholas Hud speaking next. He's a professor of chemistry and biochemistry at Georgia Tech, and he also directs the NSF NASA Center for Chemical Evolution, which is quite a well-funded large consortium that looks at a lot of issues related to chemical evolution. And he's going to tell us today about a self-assembly approach to RNA, the challenges and advantages of water. So you sound pro-water.
Well, thanks for the introduction. Yeah, in this crowd, I don't know if that's a good thing or not. As you had noted in the biological realm, it usually is. But the leading title was actually what Jack gave to my talk, and I never changed it because I thought it was very good. Is water necessary for the origin and evolution of an RNA world? And in our center and in my lab, this is a question that we really struggle with a lot. We're trying to understand the origin of nucleic acids. And one thing, if you're trying to go into this unknown realm, is to really start to question all the dogma. Do you need water? Do you not need water? And that's a question we've wrestled with. But we've also been working a lot on the assumption that life starts in water, and with that, what are the advantages and challenges? So I'll give you a little bit of background on this problem of getting RNA and why do we care about RNA? Well, there's a hypothesis, quite a popular hypothesis among original life researchers, many, which is called the RNA world. And the idea is that some polymer must have come first, and RNA looks like a really good one because it can store information and it can catalyze reactions. And this hypothesis really took off after it was demonstrated that in life today we still have RNA molecules that do indeed catalyze reactions, natural reactions within the cell. And in a way, it might have gotten a little too out of hand, where Joyce and Orgill dubbed this what they call the molecular biologist's dream of the RNA world. And I say that this is kind of an extreme view of the RNA world. And the view is that somehow on this prebiotic Earth, there was this warm little pond, so to speak, where RNA polymers somehow spontaneously formed. Now, we know that if we have a pool of polymers with random sequences, RNA polymers with random sequences, that a small subset can be catalytic. And then it really gets into the hypothesis that maybe some of those that were catalytic could also self-replicate, and if they could self-replicate and they had the ability to mutate, then they could undergo Darwinian evolution. So that's really the extreme view of the RNA world. I don't really hold to all of this for sure, but I do think that nucleic acids were important, so we want to know how they came about. So how difficult is it to make RNA prebiotically? Well, the first real, discovery that's, I would say, gave us an indication that it may be possible to make nucleic acids abiotically was back in 1960 when John Oro was looking at concentrated solutions of hydrogen cyanide. And what he found is that hydrogen cyanide would tetramerize, and this is a well-known intermediate in this reaction. If you can add another hydrogen cyanide to that, you can actually make adenine. And if you look at the chemical structure of adenine and the chemical composition, what you see is that it really is just a pentamer of HCN, which is very intriguing that our biological molecules really could connect back to some very, very basic, simple chemistry, as they should. HCN is not the only chemistry that you can use to get these molecules. There's another closely related molecule that's formamide. This is actually the single hydrate of HCN. This is an interesting molecule because it's actually a liquid over a wide range, wider than water, from 2 degrees C to 210 degrees C. And if you heat up this liquid and you shine some UV light or put some minerals in there to do catalysis, you can actually make all 4 of the nucleobases that are found in RNA. So again, this is quite encouraging to us that the building blocks can be made. And I emphasize here, plus many other pyrimidines and purines. It's not like this reaction here only produces those that are found in life. And that's a big challenge, and we'll talk about that. It's both a challenge and an opportunity. So what I'd say is that virtually all the building blocks of extant life, of extant biopolymers, the ones that we have in life today, can be synthesized in model prebiotic reactions, as well as many closely related molecules. So then comes a big question is, how did small molecules become the first biopolymers? That's something we really are striving to understand as prebiotic chemists and in our center. So this brings us to another description of the RNA world that was dubbed by Joyce and Orgel again as the chemist's nightmare. This is the prebiotic origin of nucleic acid. And RNA or DNA, these molecules to a chemist look incredibly complex if you go about to make a polymer like this. Just all the different ways that you can connect these molecules together, the nucleobases, the sugar, the phosphate, to give you one product. That's a huge challenge here, how you do that. But now think about these molecules here in what would be a, say, broad prebiotic chemical inventory. And you have yet another challenge, which is molecular selection. How did the molecules that will eventually become RNA somehow get partitioned from this wider group of molecules so that they can then selectively interact and be coupled together into RNA? These are two big problems. So we started working on this problem. I've been looking at this for about 20 years now. And what we like to do is we like to think about what are the most germane parts of these molecules that could give us clues to what came first, and then look at the paradoxes that can teach us something, or at least enlighten us a bit about alternative chemistries. And so one of these questions about selection comes up. If you have this mix of all these different heterocycles, it begs the question of how did the first bases get selected? Now, I think most biochemists, most biologists would agree that Watson-Crick base pairing is really the essence of information storage and transfer in life. It is the pairing of bases between the iconic duplex of a double helix. That molecular recognition really ultimately is what codes for all of our proteins, all of life. It's stored, right? It's transferred with these. So we would think that the bases were probably selected because they can pair. But that brings up a paradox we like to call a paradox of base pairing, which is that if you have oligonucleotides of complementary sequence and you mix them together in water, they will form a duplex. So it's a great example of molecular recognition. However, the nucleobases of present day do not pair if you mix them into water. In fact, they stack on top of each other. They don't make Watson-Crick base pairing. And some of this data actually was collected at Caltech in the 1960s by NMR spectroscopy showing that they stack up. It makes a lot of sense. It makes a lot of sense when you're in water because water is 55 molar. You got two hydrogens, you got two lone pairs on your oxygens. That's 100 molar in hydrogen bond donors, right, and acceptors. And so there's not a lot of energetics to hold these two base pairs together. These edges like to be exposed to water. And so they like to stack up, and you just make these stacks without pairing. And that's a problem then, because then how would these heterocycles have selected each other out for just pairing ones to go into polymers? If you just polymerize everything, you have a horrible mess. I don't think you'll ever get anywhere. So this brings up another hypothesis that's guiding our work, which is the hypothesis of nucleic acid evolution. What we think is that RNA actually started out as what we could call a proto-RNA polymer, a polymer that was easy to assemble. It had different bases. It had, instead of the sugar, what we call the trifunctional connector. And instead of the phosphate, it had something else that we call the ionized linker. Looked similar, had similar properties, easier to assemble, but over the course of evolution, each one of these components was changed out until we got to RNA, which is a functionally superior molecule, but much, much more difficult chemically to synthesize. And some people will resist this, that like RNA first, but I'd say they also accept that RNA, probably at some point evolved into DNA by losing an OH group here on its sugar and changing one base. And in a way, DNA is functionally superior in what it does. So if you accept that RNA evolved to make something superior to itself for a specific purpose, then why not accept that RNA has evolved from something before that was functionally inferior? So guided with this hypothesis, what we started to look at a few years ago in my lab is kind of bite the bullet and look at different heterocycles that could have been present along with the bases that we have in life today, and ask the question that could some of these nucleobases have solved the problems that we're facing with the synthesis of RNA. So these 4 outlined right here, circled, of course, are the ones that we find in RNA. I hope you appreciate that if I didn't circle those, they'd be kind of hard to find because they look a lot alike. And that's one of the points, is that chemically they are a lot alike. They're a lot alike in their reactivity. They're a lot alike in the way that they interact with water and with each other. But maybe there's some special ones in there. And the first one I'll tell you about is some work that we've done with this molecule right here, 2,4,6-triaminopyrimidine, or TAP for short. I got interested in this molecule for a couple of reasons. And one of the reasons is to answer this problem of selection and molecular recognition. It had been shown back in around 1990 by the Whitesides group and Jean-Marie Laden's group that triaminopyrimidine and its analogous molecule melamine, which is a triazine, and this is a pyrimidine, and this is even easier to synthesize prebiotically, that these 2 molecules can form pairs, base pairs, with barbituric acid and cyanuric acid. Now this is intriguing to us. Now almost all of this work was done in organic solvents, and that made sense because they were looking at these structures and saying they will hydrogen bond together. We want to maximize the energy of hydrogen bonding, so get out of water, get into an organic solvent, and that'll work. We got intrigued by this because what was also known is that if you take triaminopyrimidine and you mix it with barbituric acid or cyanuric acid, it forms a complex that precipitates from water. And to us, right, a lot of people say that's horrible, it's precipitate. But that to us meant that it was the molecules were recognizing each other and forming a stable assembly. So what we wanted to do was then explore the possibility that maybe something like triaminopyrimidine and cyanuric acid were the original base pairs. And I hope by the way that I've drawn these here, you can recognize it looks a lot like a Watson-Crick base pair. In fact, I can go through a couple steps, close up a ring here, change a nitrogen for a carbon here, get rid of oxygen, and we're pretty quickly at a Watson-Crick base pair right here. Now, as I mentioned, these form larger structures, and what had been recognized by Whitesides and Len group is that hexads are a possibility. And so maybe life didn't start with a Watson-Crick duplex, but maybe with some type of a Whitesides-Len hexad, if you will. Now, the problem that I mentioned is that in water, these precipitate because they make this kind of graphite-like structure. But what we thought about was, well, maybe there could be modifications that could actually block the formation of that structure. So as an inroad into this, what we did was a synthesis where we took this molecule, triaminopyrimidine, and we just modified one group right here. And what we did was we put on this succinic acid tail right here, give us a charge here to make it a little more soluble in water, but also what is key is that we block hydrogen bonding to this face. And so our hypothesis is that these structures would form potentially in water and stack up. And it would be that this large hydrophobic surface right here would cause the formation of a stack. So we synthesized these molecules, and the first indication that we had that this was working was that when we put triaminopyrimidine modified, that we call tapas, in with cyanuric acid into this aqueous solution, at any concentration at 5 millimolar above, what we found is it was forming a gel. And so you can see here at 3 millimolar, this is called by the cryptic name the inverted vial experiment. Okay, that's a joke. Like the simplest experiment we ever do in my lab. So you just take this vial and flip it over, and you see the liquid falls down like that. That's just like water. But if you're at 5 millimolar, it's sticking up here. This is a gel, and it's sagging a little bit. And then 15 millimolar, you can see that's a nice firm gel. Now, the reason that is forming a gel is that the monomers are forming supramolecular assemblies that are so long right? It's like in your Jell-O where the proteins are connecting in a way that they're really holding the water, freezing the water in place, so to speak. Now, these polymers are so long that when the monomers are assembled in them, they're invisible by NMR spectroscopy. If you do solution state NMR spectroscopy and you're looking at molecules, what you know is that small molecules, they tumble very fastly and you see their spectra, but if they're in large assemblies, they tumble slow and they're invisible. So I bring that up because what we do is we look at the NMR spectra of these molecules in solution, and what we've done here is we've plotted the NMR visible intensity here versus the concentration of these molecules in our NMR tube. And what you see is that you start at 0, of course, no signal, but you have a linear increase in the signal as a function of concentration, which is what you would expect on an experiment like this. But then when you hit about 3.5 millimolar, it flattens out. And what that means is that you can add more and more molecule, all you want in there, but you have no increase in signal. And the reason is that all of the additional molecules that you're adding in there are going into assemblies. And what this tells us is that these two molecules, triaminopyrimidine and cyanuric acid, have this association constant at 3.5 millimolar. But what's even better than that, this is really a phase transition. There's only two states that are possible. We have monomers soluble in solution and very large assemblies. Well, what do these assemblies look like that I'm talking about? Well, we took a little bit of that gel that I showed you and we smeared it out on a piece of mica and we imaged it with an atomic force microscope. And here's what you see. You have these nice fibers. And if you scan across one of these and you measure the height of them, it's like 2 nanometers wide. It's just what we expect from a hexad with some modifications. But what's really fun about these is that if you look at the length of these, this right here, that's a 0.5 micron here. These are pi systems that are stacked up. So that we know the thickness of that is 0.34 nanometers. So if you look at one of these fibers here that's about a micron long, that means that you have 3,000 stacked hexads, which is like 18,000 paired bases, very organized. And this is all non-covalent, yet we're getting assemblies, so to speak, that are like gene-length assemblies. And this we were really excited about because this is what you want. You want to see that from the start you could have molecules that can associate and start to make structures, right, that would lay the groundwork for making your biopolymers. So what we have here are bases that are pairing and making structures that are akin to RNA, but they're not hooked together yet. So we know quite a bit about why these assemble. It turns out that there's an enormous hydrophobic effect, as I mentioned. If you look at the surface area of one of these hexads, to expose a hexad to solution gives you something amazing like 27 kcals per mole based upon calculations from theoreticians. And so what that means is that we have a big arrow going over here that if we make a hexad, it will go into the assembly. It's about a 10 to the 10th equilibrium constant favoring the assembly over free hexads. So that's why we have a phase transition that you're either these monomers or you're these polymers in solution. Okay, so that looks pretty good that we can select out bases. So let's go back for a minute now to this nightmare that we have. So let's say that we have the bases here and we can select them out. How do we couple them together? This is yet another problem. So before it was a question of non-covalent assemblies. Now we're looking for covalent bond assembly. So really our biggest clues, I think, in the origin of life, the origin of molecules, are ourselves. Ourselves as, that is, biological organisms. We look at ourselves in our chemistry, and I think we get clues. Now, in the biopolymer structures, they really provide us clues, I think, to their origins. The polypeptides are made by, take amino acids, you stitch them together, you make an amide bond, you get a dipeptide, and off comes water. You keep doing that, adding amino acids, water comes off at each step, and you make a protein. Nucleic acids are more complicated. You have to take a sugar, connect it to the base, but when you do that, through this amino linkage, you have water coming off. Take a phosphate, connect it onto a nucleoside, water comes off, you have a nucleotide. You take nucleotides, you connect them together, each time water comes off. So each bond that I mentioned there is a different type of bond, different functional groups that are coming together, but all are formed by what we like to call condensation dehydration reactions. Condensation molecules are coming together, they're forming a bond, forming one molecule from two, and then dehydration. Water is evolved, water's coming off. So this gives us a little bit of a paradox again, which is that the best place to promote condensation dehydration reactions is in a dry environment, right? This is where I can say in a friendly cloud, the last place I would want to make biopolymers is in water, okay? Because I want to get rid of water, right? So what I can do is I can put monomers, say, on a dry surface here and heat them up or dry them down, and then they will be driven toward polymerization. But there's a problem here too, because life is believed to start in water because polymers, the polymers of life, they function in water. They fold up, enzymes fold up. And so you expect an aqueous environment. This seems like a bit of a problem. We can look at the specific bonds that need to be made in RNA, these covalent bonds. One of them is the glycosidic bond that's formed between the base and the sugar. This has turned out to be such a pesky bond to form that it has its own name. This is called the nucleoside problem. How do you make the nucleosides? How do you connect a base to a sugar? And then you need to take a phosphate, put it on the sugar. That's a phosphoester bond. That's phosphorylation. And then you need to take those and polymerize them together once you have the nucleotides. And so that's the phosphodiester bond formation. Each one of these has its challenges. And I don't think any of them have been completely solved to our satisfaction yet. So what we're looking for is there a simple, plausible prebiotic reaction that can get us through each of these bond formation. And Orgel down in San Diego and his co-workers in the 1970s really pioneered studies in this area. And they said, well, if it's easy to make adenine, and we know that we can make ribose and other sugars by other model prebiotic reactions, then is it possible that there is a simple reaction that will allow these two to couple together to make a nucleoside, forming this bond right here and evolving water at the same time? So how do you do that? Well, what Orgel explored was a very simple hypothesis. Again, where do you want to do this? You want to do this in a dry environment, but there's the problem that biopolymers want to be in an aqueous environment. That's okay. You're in a desert, it dries down, clouds come, it rains, right? Repeat the cycle. It's easy, right? The chemistry should be easy, okay? Well, devil's in the details, right? So Orgel took adenine, he and his colleagues, they put it with ribose, put it with some catalyst, heated and dried it. They got adenosine in about 1 to 5% yield. Well, that was the good news there. The bad news is that guanine really didn't work, uracil didn't work, cytosine didn't work. So this is where we're left from 1972 for about 30-some years with a problem with the nucleosides forming these other nucleosides. So again, we started thinking about this idea about nucleic acid evolution. Could we form nucleosides with other bases? Now, in the interest of time, I haven't talked about formation of nucleosides with triaminopyrimidine. That is certainly something that we've published on, and that is possible. I'm going to focus on two other molecules here, barbituric acid and melamine, for symmetry reasons, so to speak. And these are two that I introduced earlier that were also explored for their ability to form these types of assemblies, these hexad assemblies. And so what we wanted to see is it possible that there are molecules such as melamine, barbituric acid shown here that can solve two problems at once? Could they form nucleosides with ribose? Could they form assemblies like hexads and self-organize? Now, again, I just want to emphasize that even if you could form the nucleosides and the nucleotides with adenine and uracil, they will not form base pairs in water. So you got two problems here, getting these molecules to form their nucleosides and nucleotides and to get them assembled. Could there be molecules that do this together? So the first one is, does melamine make nucleosides when dried and heated with ribose? And what I'll point out here is that here's the structure of melamine. It has this exocyclic amine here. We're looking for reactivity there either by ribose or ribose 5-phosphate. And this is kind of an analogous molecule to a purine, like adenine or guanine. And in fact, if you close that ring right there, you take off this nitrogen there, then you've got an analog of adenine. And then if you switch this to an oxygen and close that ring, you're at guanine. So we're thinking maybe this is an ancestor. So we take take ribose and we heat it up. Remember, we got 1% yield with adenine. And do we get formation? And the answer is resounding yes. Turns out 55% yield if you just heat these molecules together. It's really easy. Really, really clean reaction. If you're looking at this here, this is actually unpurified sample right here. And the majority is going to nucleotides. It just wants to do it. So, and this has just been submitted. So we're pretty happy. We can make the nucleotides really well. And then the next question is, can we make these with barbituric acid, which is potentially a pairing partner when it's dried and heated with ribose? And the answer is yes. And in fact, this one's even easier. It goes at 82% yield. And again, unpurified here. Very clean reactions. Again, this is what we would hope. Prebiotic chemistry should be easy. We should be able to put in molecules that are robust and have them move along the course towards the biopolymers. Well, what if we take those molecules that we just made in those one-pot reactions, we mix them together without any purification, and here's what you see. You look at them in the circular dichroism, which shows you whether you have chiral assemblies. You get a big signal that says, yes, they're assembling in water. You put them down on the AFM, and you see that you have these nice assemblies again. So maybe we're one bond away from proto-RNA. This is where we're at. We can assemble, we can make nucleotides. Now we need to link them together. As I mentioned, there's 3 real big bonds that we got to make. We've got to make that nucleoside, we got to phosphorylate, then we got to polymerize. And that's where we're at, kind of a progress report on this. We're working on this right now. Our current goal is to do that. So in the last couple minutes, I'm just going to switch gears and tell you about also my love of non-aqueous solvents, a little bit of that. We've been looking for several years now at the possibility that nucleic acid structures could assemble in non-aqueous environments for both prebiotic chemistry reasons and also for applications, practical purposes. And an area that we've gone into is looking at, it's been mentioned earlier today, is ionic liquids. They look like they could be very promising for solubilization of nucleic acids. And we've also been looking at these solvents that are called deputectic solvents. And one that we've got a lot of use out of is made from combining choline chloride that has a melting point of 302 degrees C with urea, melting point of 133 degrees C. And that depresses the melting point of these to where they're liquids at room temperature. They melt at 12 degrees C. And this point was brought up earlier about eutectics. I think eutectics absolutely is something worth looking at. They will depress melting points, really change the properties of solvents. And they're almost inevitable to happen if you have these mixtures. Just to show you that we actually can take nucleic acids in there. I won't go through all the details on that. But nucleic acid duplexes are relatively stable in this deputectic solvent without any water around. And more than that, what we found is that not only can we make duplexes, we can make triplexes, and we can make what are called G quadruplexes, all stable in our deputectic solvent. So to me, this is very promising. It's not an absolute that you have to have water to make the nucleic acid structures. So just to end here, I'd say go back to the title that Jack gave for my talk, Is Water Necessary for the Origin and Evolution of RNA World? Well, first, I want to put a kind of a disclaimer on there and say that the RNA world is a model. Okay, we always have to be careful, right? To not get too enamored with models. And I love RNA, and I want to find out how we can make it. But it's also possible that polypeptides, polysaccharides, and lipid assemblies also played a major role in the origin of life, and that they emerged along with RNA or a pre-RNA. And some of these other polymers might be a lot easier to make, and there could be other chemistries that give rise to those. And so we have to keep that as an open mind. It's not, I don't think it's an absolute necessity for life that we get to RNA or that we get to it early. And then the question then is, water necessary for the origin and evolution of life as we know it? If we expand it out as that, that's another question. And so to answer that and to take the typical scientist way of, I'm not going to tell you yes or no because I don't know, I'd say that some reactions / processes, as I've shown you, would be easier without water. Sure, making these bonds will be easier, but some would be more difficult without water, right, depending upon the nature of the assemblies. And I'd say that in any case, life would be very different without water, right? We have to think about the nature of the chemistries that are possible. And I'll just end and say that this work it has been done by a group that's been funded by the Center for Chemical Evolution. Just some of the students have worked on various parts of the project. Most of what I've shown you on nucleoside formation was actually done by Brian Cafferty, and Irina had done started the non-aqueous solvent work, and this is done with great collaborators, in particular Ram Krishnamurthy down at Scripps, who worked on the nucleoside problem. And thank you.